am Sana Prasad and PhD scholar veterinary microbiology in III Newcastle Disease University, Rawalpindi. I am going to present a study on molecular study of circulating class II Newcastle disease virus in vaccinated broiler birds in Rawalpindi, Pakistan. As already discussed, that Newcastle is very important disease and highly contagious and list A disease that affect wild birds as well as domestic poultry. We discussed the study on broiler birds especially, and we considered and specifically validated viruses. For this study, the methodology used as the samples were collected from Rawalpindi division, including Rawalpindi district, Rawalpindi, Atak Chakwal, and Jihlam, and only from the vaccinated broiler birds with high morbidity and mortality up to 90%. On the basis of specific clinical signs and symptoms, the 72 farms, 216 samples were collected and isolated. The other steps for processing of samples, the virus was isolated in embryonic eggs and followed by PCR and meal death time assay for pathotyping, and then some selected samples were sequenced and phylogenetic tree was constructed. Following results were obtained. Prevalence or positive percentage of NDV was recorded very high in this region. In Rawalpindi, it was 80%, followed by Jihlam 71%, and Atak Chakwal almost 50%, despite of vaccination. Some seasonal data gives the result as the NDV is highly prevalent during the summer season, in which monsoon season is included. And highest percentage was recorded in Jihlam, it was 52% and 50% in Chakwal. In other seasons of the year, like winter season, the percentage was also high, but in spring and autumn season, the cases were present, but percentage is little lower than summer and winter. It means that season is the main factor that affects the outbreaks of NDV. For molecular characterization, the results showed that some of the samples were sequenced and submitted to NCBI, and they are clustered with genotype 7 virus, and that is velogenic and most important. All the samples were phylogenetically analyzed and tree was constructed, and this tree showed that all the sequences are clustered with genotype 7.2 virus, we can say subgenotype F virus, and these samples show 100% similarity with the Indian strand, Iran strand, and Chinese strand. So we conclude our study that prevalence of NDV genotype 7 is high in Rawalpindi division. NDV is prevalent throughout the year, but season like winter and summer are most important, and harsh environment affect the birds most of the time. NDV genotype 7.2 class 2 virus is circulating in the field, and this is the paper, my publication. I am concluding my session by giving the recommendation of genotype 7 or homologous local vaccine, use of homologous local vaccine to control the NDV. I am paying my thanks to organizer of conference, and also my supervisor, Dr. Aisha Riaz, and two mentors sitting here, Dr. Khalid Khwaja and Dr. Munir, Lancaster University, UK. Thank you so much.